So what I'd like to do is welcome and I also want to acknowledge my co-moderator, Reggie. You might know her. She's part of the COMMeND program as well. Maybe like she is the COMMeND program. All right, so anyway, we're going to start off with Kabi, and she's going to talk about the role of diet in tympanostomy tube otorrhea. So Kabi, come on up.
Thanks everyone for that warm welcome. Okay, can I just use this to crack them?
You can use the arrows. They're a little more reliable.
Here, okay, perfect.
Yep.
Good afternoon, everybody. My name is Kavita Dadia. I'm one of the pediatric otolaryngologists at the Children's Hospital of Philadelphia. Today I'm going to discuss the role of diet in tympanostomy tube otorrhea. I have no disclosures. Tympanostomy tubes are the most common pediatric surgical procedure, most frequently done for otitis media. But about 26 to 83% of kids that undergo ear tubes also have ear drainage, also known as otorrhea. And this could be quite impactful on patients' lives and their quality of life. And so there have been several studies that have looked at why children are more likely to have drainage. Most of the pathophysiology has been focused on biofilms, some on smoke exposure, young age, recurrent otitis media as a surgical indication, recurrent upper respiratory tract infections, older siblings, and diet. Though some of these have been named as increased risk of otorrhea, there are only a couple that could potentially be modifiable: smoke exposure and diet. And today we're going to be focusing on diet. Some of the work that's been revolving around diet and otitis media has typically been done in overall otitis media, not on the surgical population. Braccio performed a cross-sectional study in preschoolers in Connecticut to assess food preferences. So this is not what their daily intake. It looked more on what food they preferred. And they noted that children with higher otitis media rates and had lower preference for vegetables and fruit, and a higher preference for foods high in fats and sugars. Another study out of Finland performed nasopharyngeal swabs and food frequency questionnaires in children that went to daycare. And they identified that those that reported diets that were higher in sugar had an increased rate of pneumococcal carriage. There was a study from Korea that looked a cross-sectional population-based study that noted that a 10% increase in fat intake was associated with higher odds of otitis media. And then finally, animal studies and other clinical studies have also reported that vitamin D deficiency can increase the risk of otitis media, all pointing at a potential role of diet in the pathophysiology of otitis media itself. None of these studies particularly looked at the surgical management. So a couple years back, we performed a short food questionnaire to evaluate the role of diet in otorrhea. And these are all kids that had, so we looked at 300 patients with ear tubes, and we asked families about a short food frequency questionnaire about their fresh fruit, vegetable intake, fruit juices, sweet tea, diet, soda, and dessert intake, trying to get an overall picture of potential inflammatory products that the children may have eaten or drank. And we noted that children with a higher dessert intake were at higher risk of ear drainage compared to a lower intake. And these are kids that had any drainage or no, when we compared to no drainage. So kind of taking that background, we wanted to design a new study that the objective was to evaluate the role of diet quality a little bit more specific in children with tympanostomy tube placement with not just any episode, but with multiple episodes of otorrhea. And our hypothesis was that children with multiple episodes have diet that's in worse quality than those with just at most one episode of ear drainage. So we recruited in our clinics patients that were aged 2 to 6 years old enough that they would be able to tell us what they're eating, but not too old where they're making their own decisions. So that's kind of the thought process of the 2 to 6 years of age. And anyone that had the ear tubes within 6 months to 2 years of having tubes placed to capture the time where they still have the ear tubes in place. All the procedures were done at the Children's Hospital of Philadelphia. If they had an outside procedure, they were not included. Other exclusionary criteria was any health conditions that would increase the risk of otitis media, craniofacial syndrome, Downs, cleft palate, known immunodeficiency or lack of vaccination, anything that would impact nutrition, G-tube, nutritional disorders, and non-English speaking families, because some of our diet questionnaires and those were not in Spanish. The methods is, again, we recruited in clinics. Once we recruited patients, they filled out a questionnaire that had SES socioeconomic status variables, as well as other risk factors of otitis media. We also obtained the child's height and weight at that visit to help with our diet analysis. And then they all underwent a 3-day, 24-hour diet recall. This is considered the gold standard to collect nutritional information. And so how this occurred was that families were called by a CHOP research nutritionist. They were called over a course of 2 weeks on 3 separate days, typically 2 weekdays and 1 weekend day. The families were also counseled not to change their eating habits and to eat their regular diets. Our primary outcome measure was otorrhea episodes within the 6 to 24 time frame, and we dichotomized either to less than or equal to 1 episode or greater than 1 episode. Our primary predictor was estimated energy requirement percent, and this was our indicator of diet quality. Now, how we calculated this was the EER, or the estimated energy requirement, took into account the patient's age, sex, weight, height, and physical activity level, and that was set at 1.2 for all patients. And then using this, we calculated the EER percentage, which takes the average 3-day caloric intake, and we divide it by the EER. If the EER percentage was lower than 80, then they were in the lower caloric intake group. If it was between 80 and 120, it was typical caloric intake, and greater than 120 would be the high caloric intake group. The covariates that we collected, again, through both the questionnaire and chart review, were age, sex, race, ethnicity, insurance, the highest combined employment and education, breastfeeding greater than 6 months, smoke exposure, daycare or school, pacifier and bottle use, siblings, immunizations, history of premature birth, history of otitis media, or ear tube placements. These were all to be used in our analysis. So of the 137 patients that completed the survey, 120 completed the 3-day diet recall. The sociodemographic characteristics of those that completed the 3-day and those that did not were similar between 2 groups. The only variable that was slightly different was age, and it was noted that those that did not complete the 3-day were slightly older in age compared to those who did. When we look at our overall cohort characteristics, you can tell they're pretty similar between both groups. The main factor that was slightly different between the 2 groups was age at time of ear tube placement, with those that have more episodes of otorrhea were younger than those that had less. Overall in the cohort, we had more males. Most of our patients were of non-Hispanic white race, had private insurance. Most of the families were full-time employed, and most had at least a college degree. When we were looking at other factors, again, no difference between the 2 groups. So they most were breastfed for greater than 6 months. A majority did not have any smoke exposure. Most of our children went to school, did not use a pacifier or a bottle, and had about 1 sibling at home. Immunization rates, majority were immunized and not significantly premature. About 60% had a family history of otitis media, and most did not have a history of ear tubes. When we looked at the diet characteristics, average carbohydrates, vitamin D levels, and fat were pretty similar between the two groups, as was added sugar. And the main difference we identified was in diet quality. There was a difference between the two groups in diet quality, which we'll get into in a little bit. So looking at the nutrition markers in another way, looking at the entire cohort, as you could see, the 3-day average percent carbohydrate intake was within the recommended average for most of our cohort, which is between 45 to 65% of calorie intake. When we look at the 3-day average for total fat intake, most was between the 20 and 35%, but there was a little bit more of the patient population that had a higher fat intake. And then vitamin D intake, there was actually quite a bit of children that did not have the recommended vitamin D intake overall. And then the average sugar intake was about less than 50% in most of our cohort. So we performed a multivariable logistic regression, and we adjusted for age because that was a significant factor that was different between our two groups. And we noted that compared to those that have low EER percentage, those that have average or high had a higher odds of having greater than one episode of otorrhea. We repeated these analyses with accounting for the other socio-demographic variables and did not find any significant difference. So one can conclude that children who have ingested the average or higher than average amount of calories measured by EER percentage have approximately a 5 to 6 times higher risk of multiple TTO episodes compared to low caloric intake. We found that there was no difference between the two groups in looking at specific diet components, although I would like to add that overall there was a low amount of vitamin D intake in our total cohort. And this suggests that there is an association between diet quality and tympanostomy tube. Larger studies will need to be done for causal. I'd like to thank my co-authors who were instrumental in carrying out the study, Alyssa, research nutritionist, our APPs in clinic and research APPs, Jill, Ashley, and Terry, and Dr. Stallings, who is the pediatric gastroenterologist who's an expert in 24-hour diet intakes. And I would also like to put in another plug for our upcoming ISOM meeting. If you have not already submitted an abstract or thought about it, please consider submitting. It is open until March 5th, or please just attend. Thank you.
We have time for some questions, so would anybody in the audience have anything they'd like to ask? If not, I have a couple questions. Sure, I'm on here. At what age were you able to get answers from the children themselves, and when did it have to be parent supplied?
So typically probably the 2 to 3 year olds, they were not able to get very much information from the child, but they would always ask. But more, I would say like more the 4 to 6 year olds that they'd get more information.
And what about the accuracy? I mean, many of these kinds of surveys, suffer from, people who are loath to admit how much fat and sugar they have.
Yes. So the way that it was done, it wasn't the fat and sugar. They would say like everything that they ate within that 24 hours. So they would say like, what are the brand of cereal? We gave them measurements of, how much is like a cup, how much is a spoon, so that they had all this information of how to give us the information that we would need. And then those data would be entered in by the research nutritionist, and from their, like the dietary aspects of it, then they would calculate like how much was within each calories and fat and average and all the other diet information.
I knew you'd have a good answer to that.
Great talk. I always wonder about the impact on diet on just the general immunity in children in general, and I wonder if how often they're getting URIs or other kind of illnesses that might be the causal or predispose them to rotaria? Like, looked at that or measured that at all?
We did not specifically measure that, but I do know that sometimes when I am seeing patients in clinic, like one of the things that a lot of kids will have like reflux and draining ears, and a lot of it is that they're drinking milk right before bedtime. So that's something that I clinically ask. We didn't specifically, investigate that in this project.
Any other questions? You want to introduce the next?
Thanks.
Next speaker is a favorite G32 resident from the University of Colorado School of Medicine. Matt Hill is at the end of his second year of rotation for his research, and he will be talking about chronic otitis media mouse model.
All right. Good afternoon, everyone. My name is Matt Hill, and like Reggie said, I'm one of the research residents at University of Colorado. And I am in the Santos Cortez lab, and I'm here to talk to you about the A2-OMA1 knockout mouse model, and specifically hearing threshold and middle ear analysis. So a brief overview of the talk this afternoon. I first want to touch on some background and significance. We'll then dive into the ABR results. Then I'll discuss some of the gross anatomy and histology findings. And finally, we'll touch on some future directions and conclusions. So first and foremost, our lab recently published findings demonstrating that the A2-OMA1 knockout mouse serves as a strong model for non-syndromic otitis media. It exhibits hallmark characteristics such as middle ear effusion, perforation, and sclerosis. Given this, it's important to recognize the broader significance of otitis media before jumping into my data. Otitis media, it's one of the most common pediatric infections, the most common cause of pediatric hearing loss, and significant financial burden both nationwide and globally. There are known risk factors for otitis media, things like age, secondhand smoke, allergies, and daycare. But despite the maximization of public health interventions, there's still an extremely high incidence of otitis media. This points to other risk factors, including immune weaknesses and genetic predisposition. Studies have demonstrated that heritability of otitis media ranges anywhere from 22 to 74%, depending on the otitis media type and cohort. As more studies investigated genetic factors of OM, variants such as A2-OMA1 were found to be associated with recurrent OM susceptibility in humans. I won't go too much into the mechanisms of this, but just know that A2-OMA1 is a protease inhibitor. It's proposed that the protein may have overlapping protective roles in the middle ear by inhibiting proteases secreted by microorganisms, thereby preventing mucosal damage and maintaining ear health. So traditionally, mouse models for OM, they rely on inducing acute infection through inoculation of the middle ear, for example, with something like non-typable Haemophilus influenza. We have few genetic models with spontaneous or chronic OM. Our lab acquired a CRISPR-Cas9 knockout model due to those previous studies demonstrating A2-OMA1 knockout associated with increased incidence of OM. We then went on to demonstrate the increased prevalence of otitis media in A2-OMA1 knockout mice in comparison to wild-type mice via histologic and gross anatomic analysis, looking at things like TM perforation, sclerosis, and effusion. Moreover, the study demonstrated there's no skeletal abnormalities typically associated with syndromic chronic otitis media. In addition to that, the average age of these mice in our initial study was on average 42 weeks. To further delineate how A2-OMA1 knockout impacts hearing, I started looking at mice less than 20 weeks of age that hadn't experienced any presbycusis or age-related hearing loss. So as a brief reminder, ABR, or auditory brainstem response, it's a non-invasive electrophysiological test. It's used to measure neural activity in response to sound. It evaluates how sound signals travel from the auditory nerve to the brainstem, and it provides insight into hearing function by providing distinct peaks in neural activity reflecting points along the auditory pathway. So my main objective was to determine if A2-OMA1 knockout mice less than 20 weeks of age develop chronic otitis media that causes measurable hearing loss on ABR. Again, focusing on age because chronic ear infections worsen over time typically. Older mice are expected to show more severe hearing loss, and hopefully once we have enough animals tested, we can compare ages, which would help pinpoint when hearing loss starts. Again, our model's advantage is that it's a spontaneous, non-inoculated model for developing chronic otitis media, and therefore there's no other external infection variables that we deal with. Diving into the ABR results, these are mean ABR threshold by group and frequency, looking at wild-type mice and homozygous knockout mice, with blue representing wild-type and red representing homozygous knockout. There was no significant differences across frequencies, but we started to notice trends, particularly in the 4 kilohertz range. This is interesting because we found mostly hearing loss in the 4 kilohertz frequency for patients with A2ML1 variants as well. Thus far, we've tested approximately 15 mice, and they've been on average 17 weeks. I mentioned not statistically significant yet because when performing a power analysis using human data as a reference, we calculated that we need approximately 30 mice per group required for ABR testing to get an adequate sample size. After performing ABR, I would dissect the middle ear bulla, and these are some representative examples of what I saw in homozygous knockout mice, things like perforation, effusion, and sclerosis. These findings were confirmed on histological analysis. This is looking at prevalence of factors related to otitis media in wild-type mice compared to homozygous knockout mice, things again like sclerosis, TM perforation, and effusion that were statistically significant at an increased rate in the homozygous knockout mice. Although our power analysis indicated that a larger sample size is needed to detect a statistically significant difference in ABR threshold, I was still somewhat surprised that we didn't observe a more pronounced difference in our initial testing. It was especially unexpected given the substantial gross anatomical and histological differences between the wild type and homozygous knockout groups. This discrepancy led me to look at the inner ear pathology if there was anything contributing to the lack of clear ABR differences. However, when looking at things like inner ear hair cell morphology, any type of blood products or effusions or anything like that, there was no significant difference. So moving forward, I want to expand ABR testing. If conductive hearing loss is present, we anticipate elevated thresholds that are consistent with middle ear dysfunction. In addition, I'd also like to look at things like laser Doppler vibrometry analysis that would demonstrate things like reduced TM and ossicular movement, which again could confirm this conductive hearing loss phenotype. In addition to the LDV, I'm also planning on performing DPOAEs and tympanometry in these mice to again get a better understanding of hearing function. So some conclusions and takeaways. ABRs do show lower frequencies are more affected, though more testing is needed in these mice. Middle ear pathology was observed microscopically, and histology showed significant structural changes in the middle ear and not in the inner ear. Again, this is validating our model that functional deficits and structural abnormalities support validity of A2ML1 knockout modeling for chronic otitis media. Limitations of the study thus far are unexplored mechanisms, things like microbiome influence and molecular mechanisms, which we again hope to explore with future experiments. Other things that we've kind of thought about thus far is why aren't ABR thresholds dramatically different? Again, we need more mice, but also maybe early stage OM may not immediately cause measurable hearing loss. In addition, there could also be compensatory mechanisms that could be maintaining that hearing sensitivity. And then why do trends appear at the 4 and 32 kilohertz range thus far? Maybe this conductive pathology may impact specific frequencies first, or we just need to do more detailed frequency analysis to reveal those hidden deficits. So key takeaways, again, the A2ML1 knockout mouse is a great non-syndromic spontaneous chronic otitis media model. Auditory function, it shows emerging trends towards impairment, but we need to do more testing. And then clinical implications, hopefully we could test anti-inflammatory treatments or mucosal repair strategies in this mouse model. I would like to give a huge thank you to the Santos Cortez Lab, Dr. Santos Cortez and Helen Gomez, in addition to the Green Lab, the Tolan Lab, and all of our funding sources, in addition to COMMEND. And I'm happy to take any questions now.
question. Have you tested have you performed microbiology tests to verify if it's a sterile inflammation or if it's an infection that has been developed?
We haven't yet. That's something that we hope to do in the future too.
Oh,
Yeah.
Nice work, Matt and Reggie. I bet you your DPOAs would be off, as I know you're planning to test them.
Yeah.
The ABRs may not be picking up some of those subtle changes. When we did our work, we would see synchronic sclerosis, some holes, minor effusions, and the ABRs would be preserved.
Yeah.
I think that'll be your next one.
Yeah. Thank you. Yeah.
I'm not as familiar with this, but is there any way to measure bone conduction in this? Bone conduction in here?
That's a good question. I'm not positive. Reggie, do you have any? I'm not sure actually. I haven't looked into that so far. That's a good question though. I'll look more into it.
I have seen some bone conduction studies in mice. I'm not sure how good they work, but yeah, I think it can be done.
And I can answer that. John Zowski published a bone conduction model in mouse. It means he had to literate the other ear. It's very hard to do if you have otitis media and preparation. It's very confusing. So it's a work in progress.
Oh, okay. I see. I'll have to take a look at that study too. Yeah. Thank you.
So our next presentation is from ARO, who's from UC San Diego, and she's going to talk to us about single-cell RNA-seq and also about microfluidic models for home study.
Good afternoon, everyone. Thank you for the opportunity from COMMeND to present some of the work in our lab. My name is ARO, and the title of my talk is Otitis Media, Single-Cell Transcriptomics, and Microfluidics Disease Modeling. I'd like to first start by disclosing that some of the research here is supported by the NIH. However, the funding sources were not involved in the study design, work, or preparation of this presentation. And so my lab works on otitis media, and we have used animal models to look at the middle ear inflammation, mainly in acute infection using NTHI. And so we've worked with knockouts, some inbred strains. We also have this engrafted humanized mouse model system in which these mice are chimeric, and they have been engrafted with human hematopoietic stem cells, where we can look at the behavior of the mucosa from the mouse versus the human epithelial cells. We also recently started to work on generating an in vitro model of the middle ear. So we wanted to design an organ and a chip that is a three-dimensional structure system that can represent the middle ear. And so these devices would allow us to utilize or maybe utilize some human specimens that we were obtaining, like from surgical procedures. And so to do this process, we assembled a team of a multidisciplinary team of surgeons who were providing us with the middle ear mucosal tissue, nanoengineering experts from the University of UCSD that had some experience with organ and chip models. And then also we had data and bioinformatics scientists, as well as cell biologists who were working with a 3D scaffold like in the lab. And so the objective of my talk and the study is basically talk about some of the work that we have done in looking at the normal human middle ear mucosal cell types and compare between the species of the mice and the human. And then at the same time, talk about the development of the middle ear on a chip model and how it can be used for studying otitis media progression. And so single cell sequencing can be used for the cellular characterization and getting a total landscape of different cell types that are present. And so we employed a 10x genomic chromium approach to look at the multiomics analysis of the middle ear. And so we were trying to identify the distinct cell types, trying to kind of look at the cell type functions, and then looking at the intercellular communications between the different cell types. And then, so here is basically a PCA cluster of the human middle ear using the 10x multiome. And you can see that we have 10 distinct clusters in the human middle ear. Major one is, I don't know if you could, is the mouse pointing, or should I use a pointer? I don't know if this is working.
It looks like the mouse. Will may not be good
Or that pointer?
Slide your mouse over to the right, and then you'll have it on the screen
Oh,
there.
got it.
Yeah.
Thank you. Thanks. Appreciate it. So yeah, so you could see the different cell types that are present and the different cell markers that are there. So we have some ciliated epithelial cells, basal cells, some endothelial, parasites, stromal cells, and then a little bit of the resident monocytes that were identified in this map. And so next, we tried to do a homology comparison between the human and the mouse. And relatively speaking, most of the cell types that were present in the human were, because we did the data, you saw probably Alan's presentation on Sunday. So we did the mouse first, and then the human was done after. But so there was a great homology between the two. But then when we looked at the specific cell, the specific genes within each specific cell subtype, we saw some subtle differences. So you see to your right, the violin plots, which visualize the distribution of the gene expression levels in the different groups of cells. And you see the differences between the mouse and the human. And we specifically focused on the HIF1 alpha VEGF pathway and the TLR and MIghty88 IRAC pathways, because they played, from our previous studies, we found that they play a key role in the immunological response. And as well, they were highlighted when we did the pathway analysis in terms of they were highly different between the two species. And you can see that only TLR2 actually right here is the one that is equally expressed between the two. It's expressed actually in the monocytes. And so when we dived in further to look at the cell-cell interactions, and for that we used actually cell chat analysis, which helps map ligand receptor interactions. And so what you see here is actually the different cell types. And so these arrows point to the senders and receivers. And so you can see that actually the basal cells by weight and size are actually the hub for a lot of the cellular interactions. So they interact with the monocytes. The secretory epithelia also play a big role because they're one of the bigger senders. They send a lot of signals into the monocytes, a little bit into the basals as well. And so when we did an overall pathway analysis, we identified that epiregulin and the HPEGF pathway were primary interactors, and they were playing which plays a key regulatory role in mucosal hyperplasia during otitis media, even from our previous mouse studies.
And so these pathways are kind of our focus in future studies looking at maybe therapies and things like that. And so our goal right now is to look at these cell-cell interactions maybe in tissue that is coming from diseased individual, which is our next step. And so finishing talking about the single cell and kind of shifting gears into the ear on a chip model. So knowing what cell types we had in the normal human middle ear, so we kind of knew what the system is there, and now we wanted to devise or create this device that would mimic a middle ear in an in vitro system. So you see here a schematic of the model. And so on the top chamber would be the middle ear. Then we would have the mucosa, and then the vasculature at the bottom, and then we would like create a flow to mimic like leukocyte flow. And so here, I think there's more. These are more pictures of the device how the device was created. It was made from transparent PDMS silicone. And so this is how like we measured the size of the opening to kind of where we were gonna place the middle ear cells. And then this is, I think, where we just tested the device prototype for like flow and leakage and things like that. And then this is just a schematic showing the prototype, like a cross-section of how we modeled this to be. So on the top, you would have the middle ear chamber. This would be seeded with middle ear epithelial cells and stromal cells. Then we had the semi-permeable membrane. And then at the bottom, we would have the GFP positive HUVEC cells. We labeled them with GFP because that was just, it would make them easy. It would make that easy to be visualized, so we could distinguish them when they get seeded, and would have a more transparent model like for visualization by like live microscopy. And then we would be introducing the isolated PBMCs at the bottom. And so this is I'll walk you through just like how the device was prepared. So here you have an image of how the device was being seeded with the GFP positive HUVEC cells. Next, on the right, you see the human middle ear explant being placed, like it's a bright field, and then this is, I think, a cross-section of both, where we have both GFP and the bright field, and the cells are there. And then we introduced an NTHI infection through the top chamber, and then we labeled the BPMCs fluorescent red using a viable cell tracker, so that it would be easier to kind of track these PBMCs into the system. And so here you see the vascular chamber, where you see the green HUVEC and then the red PBMCs. This is fluid recovered from the top chamber, where we saw basically PBMC migration into the top chamber. And then that whole thing was actually, the whole device was then fixed in PFA, disassembled, and then cross-sectioned and prepared for H&E. And here you have a control device where you see a very thin layer of the mucosa versus an OM challenged device, or like a mimicked OM challenge for 3 days, where we observed mucosal hyperplasia. Yeah, so in summary, I hope that what you got from my talk is that human and murine middle ear share a number of, or they share a lot of homology in terms of the cell types, and animal models can reveal a lot about the human immune response. But then looking at the specific genes is also important in a lot of our studies, and that's what we're hoping kind of to elucidate from some of the models that we're trying to develop. And at the same time, right now, our future direction is actually to utilize single-cell RNA-seq to profile the cell population in the middle ear on a chip after OM challenge, in addition to actually uh, pediatric patients. Um, and then, yeah, that's it. I think I would just like to acknowledge the labs that I've been working with. The Chen lab in the nanoengineering department at UCSD, Rick Friedman's lab, some of the fellows that have helped me with the collection of the mucosal tissue, our bioinformatician team at the Center for Epigenomics, and the Center for Computational Biology at UCSD, and IDCD for funding. My colleague Jason Chen, who's also been helping me with collection, or we've been working together in the pediatric patient collection. And then COMMeND for giving me this opportunity. I'm happy to take any questions. Thank you.
I have a question. That was really fantastic, Arwa. You really have to get the word out about charging your model and improve our results.
Thanks.
So
Thank you.
it is an epithelial model, though. Would anyone want a model that would also have some form of immune cells that are included in it?
So currently, you're right. We have the explants. We could try to dissociate the cells and then um, uh, do like a 3D printing of or seeding of these cells specifically. I think as we develop, like refine, because this is like a prototype one. So as we refine the model, probably that would be something that is possible or doable.
Questions from the floor?
Hi. Thank you. That was awesome.
Thank you.
Pretty wild that you can get all that happening in the one chip. That's pretty cool to see. I wonder if you compared like the mouse cells to your model cells. Do you find that there are any age-related changes in the mouse cells?
Well, these are coming from like adult mouse.
Yeah, so I wondered if you've looked at younger mice and whether that's replicated. Because I mean, a lot of your otitis media is happening in pediatrics, so I wonder if there's any difference in the tissue makeup there.
It's not something we tried, or I tried, but we could definitely look at that and see if that would make a difference. Thanks for that point.
Yeah.
No, we haven't. Yeah.
Our next fantastic speaker is Stephen Shaw. He's from the lab of Sarah Park from the University of Colorado, who is my neighbor in seventh floor of RC2. Stephen will be talking about the interferon response to acute otitis media due to streptococcus pneumonia.
Thank you. I got to press play. Awesome. I have to move this up just a little bit. Oh no. There we go. Okay. Hi. Thank you everyone for coming this afternoon. As Reggie said, I'm Stephen. I'm a PhD candidate at CU Anschutz in Sarah Clark's lab, and I'm going to be giving my talk today on the role of type 1 interferon in Streptococcus pneumoniae invasion in the middle ear. And as she had previously said, I'm going to be focusing much more on acute otitis media. So our lab, again, there's over 700 million global cases of acute AOM every year. Majority of these cases are going to be focused in on children under the age of 5, with a burden of about $4 billion in the US per year alone. So this is a very important area of study. And our lab has a mouse model that we do with an intranasal infection to then study acute otitis media. And our lab is particularly interested in the URI, the upper respiratory viral infection enhancement of bacterial otitis media, where a lot of the literature has basically described these upper bacterial acute otitis media as a complication of upper respiratory viral infections. And because of that, our lab is really interested in studying the type 1 interferon system. This is induced when you have a acute viral infection, bless you, acute viral infections, and when the virus enters into the host cell, it's then detected by your pattern recognition receptors. These include TLR3, 7, 9, RIG-I, MDA5. These will all lead to a signaling cascade that will induce the secretion of type 1 interferon, and type 1 interferon will then signal in an autocrine and paracrine fashion through the IFNAR receptor. And so our lab has a couple ways that we have gone about inducing interferon and then blocking interferon signaling to really study its importance in this. And we can induce it through using poly-IC, which is synthetic double-stranded RNA that will trigger TLR3. We can also just use recombinant interferon alpha and beta to just directly signal through the IFNAR receptor, or give a live viral infection. Our lab has used influenza A virus to give our live viral infection. It's a well-studied mouse model using influenza A. And then to block type 1 interferon, we have an interferon receptor knockout mouse line. So this lacks IFNAR1, so IFNAR1, IFNAR2 dimerize to form the IFNAR receptor, as well as just using anti-IFNAR antibodies to block that. So to start off, I'm just going to go over some data that our lab has done and also with the collaborators showing that poly-IC itself is sufficient to enhance all three bacterial acute otitis media pathogens. And so again, as we've done this, this is a mouse model where all of our mice start off with an intranasal infection of one of these three strains of bacteria, and then they are given poly-IC. All intranasally, and then we look through the surgical removal of the middle ear at the amount of CFUs. So what you see on the left side of the graph is CFUs per middle ear, and what we see is in all three of strep pneumo, Haemophilus influenza, and Marcella, that poly-IC is sufficient to enhance the infection, and basically showing that this is a model we can use to study the enhancement of acute otitis media. So my overall project goal, again, is to really identify the impact that viral-induced type 1 interferon signaling has in specifically strep pneumo invasion of the middle ear. And the first aim I'm going to go over today with everyone is determining the impact that type 1 interferon has on neutrophil-mediated clearance of strep pneumo in the middle ear. And to zero in on that, I'm really going to look at why I'm focusing on neutrophils is that middle ear infections have been characterized within the literature and our own lab with this influx of neutrophils to the middle ear. And this is important as neutrophils have both antimicrobial protective responses, but they can also have this double-edged sword where they can be very damaging and highly inflammatory. And so I'm going to focus just specifically on just two activities that neutrophils can do, which is phagocytosis, as well as the neutrophil extracellular traps, and then just to go over what you see on the left side of the slide is just gating via flow cytometry on neutrophils Ly6G high by CD11B high. And so the next data slides that we'll see will be my flow cytometry on mouse middle ears with this gating on these neutrophils. So to zero in on phagocytosis first, we really wanted to look at how type 1 interferon signaling is affecting neutrophils. And so we focused in on neutrophil production of TNF, as there is literature showing that TNF production is associated with priming neutrophils for phagocytic function. And what we see is that both as a total and as a percent, when we give poly-IC to our wild-type black 6 mice, the amount of TNF that they produce is significantly decreased in both cases, and that then when we put them into our interferon receptor knockout mice, the TNF production is restored in these mice to what we would see in the mice that have not been given poly-IC. And this is important because then when we look at our infection burdens, what we see is that the mice that have higher levels of infection have a significantly lower amount of neutrophils that are producing TNF. And the mice that are cleared have, of course, then a higher, significantly higher number of neutrophils with this function. So then our lab combined this with an assay where we have FITC labeled strep pneumo. So then we can then, by flow cytometry, look at the neutrophils that are positive for this FITC labeled strep pneumo to look at phagocytic function combined with our intracellular flow cytometry to look at TNF. And what we see is in both the groups without poly-IC for TNF production and for the positive for the FITC labeled strep pneumo, those are both higher in the groups that don't have poly-IC than the groups that do have poly-IC. So the interferon signaling induced by poly-IC is reducing both the TNF production and the phagocytic function of these neutrophils. You can then see the R squared of 0.8 showing that there is significant correlation between this relationship. So next I'm going to then move on to the other aspect of the story where already type 1 interferon is showing decreased phagocytic function in neutrophils. I'm going to now move to netosis. I'm moving to NETs because there is a significant presence of NETs found in otitis media. And what NETs are, just to briefly explain the really quick schematic that I had made on the left side of the slide, is a process where neutrophils will release their DNA into the extracellular matrix, particularly coded in antimicrobial peptides, different proteases, as well as coded in NPO with the purpose of killing bacteria. This is, however, also a very highly inflammatory event when they release their DNA into the extracellular matrix. And so we decided to focus on getting PAD4 knockout mice. And we got PAD4s because PAD4 is peptidylarginine deaminase 4, and it's involved in both the ROS dependent and independent formation of NETs. And what it does is it is involved in the decondensation of the chromatin, and this is an irreversible step in netosis. So with our PAD4 knockout mice, we put this into our mouse model and giving poly-IC to them and really wanted to look at how impactful the loss of the ability to create NETs is. And what we found again is there's a significant reduction in CFUs in the mice that no longer have the ability to produce NETs in our PAD4 knockout mice, showing that NETs are contributing to the disease phenotype that we see through type 1 interferon signaling. So just to wrap up, what we see with the role of neutrophils is that there's a decline in their phagocytic function and a potential role also of interferon in affecting NETs. So that's going to move me to the second aim that I have for my project, which is really looking at the impact of type 1 interferon on the myeloid cells from what the epithelial cells are with an influenza A virus co-infection. And to do this, I made bone marrow chimeric mice in our lab where we took CD45.1 recipient mice and then injected them with CD45.2 donor bone marrow from either black 6 mice or our IFNAR mice after being lethally irradiated. They're then given 8 weeks to recover. 4 of those weeks they are on antibiotics. And then we, at day -3, we gave them an influenza infection, and then at day 0, they were given a infection with a strain of strep pneumo, serotype 2. And what we see with that is that grouping them by the mice that got the wild type black 6 bone marrow, and then the mice that got the IFNAR bone marrow, we see that influenza A was able to significantly increase the infection burden as seen in CFUs per middle ear. And we don't see that same significant increase in the IFNAR bone marrow group. And then just to zero in specifically on the groups that were given the influenza co-infection, using flow cytometry, we further investigated the role of what impact this is having on the neutrophils. And once again, we see, like with the data I previously just presented with the poly-IC, that the wild type group with influenza A sees this significant decrease compared to what we have with our interferon, the bone marrow from the interferon knockout mice, in this decline in neutrophil TNF production and in their ability to phagocytose the FTSE-labeled strep pneumo. So once again, we have this decreased phagocytic function in the phenotype and an increase in burdens of strep pneumo in the middle ear. The current working model that our lab has with this then is these upper respiratory viral infections are leading to the secretion of type 1 interferon. This then results in an altered immune profile, resulting in a decrease in neutrophil phagocytic activity, a potential increase in neutrophil mitosis, resulting in increased bacterial burdens in the middle ear. And if we block this signaling, what we end up seeing is increased phagocytic function and a decline in CFUs found in the middle ear space. And with that, I would like to thank my lab, my mentor, particularly Sarah Clark, and I'd also like to thank COMMeND for the opportunity to give this talk to everyone today. And I will take any and all questions people have. Thank you. Cool, I did a great job then. Microphone.
Great job. With your co-infection model, so you first infect with the virus minus like 3 days, then you introduce the bacteria.
Correct.
Do you check that the virus has been cleared, or do you know that you ever check for the titers of the
So
virus?
we have collected 4 viral titers. To be honest, they're sitting in our refrigerator. We have not run them yet, but we do collect for the viral titers. We collect for the viral titers not in the nasal pharynx, though we have collected in frozen nasal pharyngeal fluid. We're really checking to see if they're present in the middle ear, though based on just talks with Alan, and just what we've just done, just giving poly-IC alone, like that's enough to change just what you're doing in the nasal pharynx, just to, the entire area, just the immune response is altered.
Thank you.
Thank you.
I had a question about the influenza infection model. How many days do you wait to make sure that the mice are actually experiencing influenza?
Oh, that's a great question. We take weights every day from the mice. I'd have to take a look at the data itself to particularly know, but we do see the weight drop to know that they do have an infection, especially compared to the control group that just gets PBS, where they have no weight loss over those 3 days. I don't know at what day we particularly see the weight drop, and it can probably vary from mouse to mouse.
Oh, I'm sure it does. I'm just, in our experience, when we, inject influenza into the nose, we don't get a true infection with every mouse. And, we wait about, 5 or 6, 7 days, and then if you see a significant drop, we know that it's taken. We're using, PRA, it's mouse adapted. But, even then, we didn't see 100% infection. So you've been getting, pretty good percentages, hopefully better than ours.
Yes, we're also using X31, but also I think the other thing to note is that the bone marrow chimeric mice are not the happiest in general anyway, and they seem to take pretty well to the infection. There's another member of our lab who's doing the infections with the influenza infections without bone marrow chimera, so I'd have to really want to compare the notes then between how successful my infections with the influenza versus hers were, because her mice are healthy and happy before the infection.
All right. Any other questions from the audience? If not, thank you, Stephen.
Thank you.
Very much. So our next presentation is from Ellen Gomez from University of Colorado, and she's going to be telling us about the role of MUC5B overexpression in the middle ear pathology and susceptibility for vaccines.
Sorry. Okay. Oops.
Hi everyone. Oh, My name is Helen Gomez. I'm from University of Colorado. I'm a PhD candidate in the Santos Cortez lab. And today I'm going to be talking about the role of MUC5B overexpression in middle ear pathology and susceptibility to otitis media. So MUC5B or mucin 5B is a glycoprotein found in chronic otitis media fluid. As we know, these mucins are responsible for the gel-like characteristics of mucoid middle ear fluids. MUC5B actually compromises as the predominant mucin glycoprotein in these effusions, and it is believed that MUC5B may be the reason for the viscosity of these chronic otitis media fluid and why Eustachian tubes do not always work. MUC5B actually plays a role in idiopathic pulmonary fibrosis, as it is localized to the distal airway epithelium. And as you can see in the first panel, you see stem cells repopulating bronchioles and alveoli under normal conditions compared to that of a fibrotic model. So in the lungs, loosely associated mucus is responsible for trapping inhaled particles, including bacteria, while still allowing transport out of the airway by ciliary and cough-driven forces. However, when this mucus is overproduced, excessively secreted, or can be overly viscous and adherent, materials that need to be eliminated actually accumulate and exacerbate disease states that can result in cellular dysfunction. And so excessive production of MUC5B actually impairs cells that attempt to regenerate any injured airway of the lung, disrupt normal reparative processes, resulting in chronic fibro proliferation and the formation of these honeycomb cysts, which are actually hallmarks of IPF. There is a MUC5B promoter variant that has been associated as being the dominant genetic risk factor for the development of IPF. Actually accounts for 30 to 35% of the risk for developing IPF, with a minor allele frequency of 8% across multiple populations. And so knowing that this variant leads to an overexpression of MUC5B, it has been studied that MUC5B actually impairs mucociliary function in mice, causing excessive retention of inhaled substances, as I said, such as air pollutants, any chemicals, microorganisms, and over time, this lung injury leads to scar tissue and persistent fibroproliferation, resulting in IPF, and as a result, forming these honeycomb cysts within the lungs. And so we were interested in looking at the association between IPF diagnosis and history of OM. We ran a multivariate logistic regression analysis where we were able to see an inverse association between IPF diagnosis and history of OM after adjusting for ethnicity, sex, history of smoking, age of diagnosis, which is suggestive of a protective effect on otitis media susceptibility, but could be confounded. We then did a transmission disequilibrium test, or a TDT test, where we looked at a total of 803 families from the UK, Finland, and the US, where we actually noted an increase in non-transmitted versus transmitted, which again might be suggestive of a protective effect, but this was not significant due to either small effect size and or small sample size. Moving on to looking at the alpha diversity, we looked at the microbiota profiles of Coloradan children with otitis media and carriers of this specific variant, and we were able to note significant differences across richness, evenness, and overall just diversity within the middle ear. However, these changes were not seen within the nasopharynx, so we know that these differences are really true to the middle ear. We then looked at the beta diversity in these Coloradan children, where we were able to see more diverse bacterial profiles in children that were carriers for this specific variant. You have a decrease in Haemophilus and Moraxella, but you have an increase in specific proteobacteria such as Lactobacillus and E. coli. Now looking at MUC5B expression after non-typable Haemophilus influenza inoculation. So this was done in wild-type mice by the Dr. Alan Ryan's lab at UCSD, where we were able to see that there's a significant upregulation in MUC5B in day 1 and day 2, with a significant decrease at day 5. And it's important to note that single-cell RNA-seq data also showed an upregulation of MUC5B in epithelial cells within these wild-type mice. There is a MUC5B knockout model that has been studied, and within this publication, you're able to note that there's some colored middle ear effusion with a retracted eardrum. You also have CFU counts where you have an increase in the knockout model showing a polymicrobial infection and also an increase in inflammatory cells. So showing you wild type mouse data, this knockout mouse model, we're interested in now looking at the CSGB1A1 MUC5B transgenic mouse, which actually is an overexpressing model. There has been no otitis media study done in these mice yet, and we're interested in studying because it's mimicking the effect of the variant that we were studying, so overexpression of MUC5B, and we want to be able to see whether it will increase or decrease the presence of otitis media based on MUC5B expression. And so we're planning doing these NTHI transbollar injections, comparing that to just saline injections, and then moving on to running qPCR, histology, immunohistochemistry, and again, getting that exposure of the boli during surgery, doing automicroscopy to confirm the injection was done and notice any otitis media hallmarks through microscopy. Here I'm going over some histology, so just showing you wild type MUC5B mice, you see organized epithelia, and then comparing that to these overexpressing mice, you kind of see that this organized epithelial layer. We have this inoculation with saline 1-day survival, again, seeing that organized mucin now, but again, comparing that to the overexpressing MUC5B mice, you kind of have that disruption of that mucosal layer and mucosal thickness as well. So currently, we are working on validating MUC5B expression in the middle ear via immunohistochemistry. We're also working on NTHI inoculations in the transgenic mice. Some limitations to know within the study, we are looking at a short-term infection model, so it might not reflect chronic otitis media scenarios where infection lasts longer than 5 weeks. Also, it's important to note that these MUC5B mice do display features of IPF, so there have been some difficulties with live surgery and survival. So kind of focusing on different age regions might fix this issue. But with that, I'd like to thank the Santos Cortez Lab, Matt and Reggie that are here today, everyone that has been a part of this study, and COMMeND for being here. Thank you.
So we have time for questions. Any questions from the audience?
Hi, great talk. I just wondered if you would see the same upregulation of MUC5B with other OTIS pathogens, whether you looked at that at all or just NTHI?
We've only looked at NTHI, yeah.
And have you looked at all in the nose for this MUC5 expression? I'm not sure, you might have said it already.
In the nose?
Yeah, in the nose.
No, I haven't looked at the nose, no, only the middle ear. Yeah.
Oh, thank you.
Thank you.
Great talk. I was wondering if you have or are looking into any of like proteomic changes of the composition of mucins.
I haven't looked at the exact genus of, yeah, I can't tell you that. I only have like the overall, yeah, but that'd be more interesting to look at once I actually have a better idea of what's going on.
Yeah, definitely let me know when you do that.
You mentioned that the surgery and the survival is kind of a challenge.
Yeah.
What is the main problem that you face?
Yeah, so we think that because these mice are used for lung disease study, they already have like a lot of like scar tissue and like scarring within their lungs. So what we notice is that they're not able to metabolize ketamine or xylazine. They don't wake up from surgery, and we've played with a lot of dosaging. Me and Matt have done a lot of surgeries, and we're only sticking to isoflurane. But yeah.
Because I have worked with a model and we have problems
Oh, we should talk about that. Yeah. Thank you.
I had a question also. One thing we note in MTHI infections is that there's not a lot of generation of frank goblet cells. But in these overexpressing mice, do you really see true goblet cells?
Yeah.
In mucosa?
Yes, we do. But we want to do specific goblet cell staining. That's also next to be able to confirm that there are goblet cells.
Okay, have any other questions? If not, thank you so much
Thank you.
for the talk. Can we move on?
Right, we're going back to Philly. The next speaker is Mary Marino from Heart Earling's lab. She will be talking about dark matter regarding MTHI. We're really excited to hear about.
Hi, everyone. As Reggie said, my name is Mary Marino. I'm a PhD candidate in Dr. Garth Ehrlich and Josh Mill's lab at Drexel University College of Medicine. As is the nature of science, I've since disproven some of the data that I planned to show you today. So my title has changed, and instead I'll be talking to you about crosstalk that I've discovered between starvation responses in Haemophilus influenzae. Otitis media, or OM, is the #1 reason that kids go to the doctor, that kids are prescribed antibiotics, and it's actually the second most common reason that kids undergo surgery, but only after circumcision. Most ear infections will resolve just fine on their own or after a round of antibiotics. Our bigger problem is when the infection isn't cleared or recurs multiple times. About 25% of kids that experience an ear infection will have this kind of persistent infection. And if the infection occurs during a critical period of language acquisition for a child, this can result in delayed language development. In some rarer cases, persistent infections can also lead to permanent hearing loss. There are a couple of organisms that can cause OM, but nowadays, thanks to the pneumococcal conjugate vaccine, most of the time it's caused by Haemophilus influenzae, or H. flu. H. flu is capable of causing persistent OM through several different mechanisms, but the two that I'm going to be focusing on today are evolutionary adaptation and the spread of resistance through horizontal gene transfer, as well as physiological tolerance. Unfortunately, about 25% of H. flu strains encode a gene that confers resistance to beta-lactam antibiotics, which are our first line of treatment against OM. The beta-lactamase enzyme breaks down beta-lactam antibiotics before they're able to inhibit bacterial cell wall biosynthesis. And we see this in the lab. When I treat a beta-lactamase negative strain with a beta-lactam called ampicillin, the minimum inhibitory concentration of antibiotic that is needed to kill it is very low. But when I treat a beta-lactamase positive strain with ampicillin, it's genetically resistant, so the MIC of antibiotic needed to kill it is over 300 times higher. And the problem of genetic resistance is worsened by the fact that H. flu is naturally competent. Under certain conditions, H. flu can express a DNA uptake apparatus that allows it to take in free-floating DNA from its environment and recombine it into its genome. And I can induce this in the lab by starving my cells and turning on this competence machinery. If I feed them DNA carrying an antibiotic resistance marker, we see that they can take up this DNA, incorporate it into their genome, and now they're able to survive on an antibiotic plate. This is how antibiotic resistance genes can spread, including the beta-lactamase gene, between H. flu strains as well as other members of the Pasteurellacea family. Thankfully, we have a beta-lactamase inhibitor called clavulanate that we often use as an adjunct to make beta-lactam antibiotics effective against strains that have acquired the beta-lactamase gene. So if you've ever prescribed or been prescribed Augmentin to treat a bacterial infection, that's what you're getting, is a beta-lactam called amoxicillin with the clavulanate adjunct. And we see this work in the lab too. When I treat a beta-lactamase positive strain with ampicillin and clavulanate, the beta-lactamase activity is inhibited, and the MIC needed of an antibiotic needed to kill it dramatically decreases. And that's good, right? Well, it's good for now, but sadly, aside from the beta-lactamase gene, there are other non-enzymatic forms of resistance that are emerging that are making beta-lactam antibiotics ineffective. So eventually our clavulanate trick is going to fail too. In addition, even if we switch antibiotics, resistance will emerge to those antibiotics as well, and it will spread through natural competence. So we have a real problem on our hands, and that's not our only problem. Now I'm going to talk to you about a second mechanism that leads to treatment failure for persistent infections, and that's tolerance. Tolerance and resistance are not the same. While resistance to antibiotics is encoded within a bacterial cell's DNA, tolerance arises independent of whether or not a cell acquires an antibiotic resistance gene. Cells become tolerant to antibiotics when they enter a state of metabolic dormancy and they stop actively dividing. So if you're a dormant cell and you're not actively making a cell wall, a beta-lactam simply is not going to work. And we see bacterial dormancy in nature in many forms, but often, in the case of disease, we see it in the form of a biofilm. Biofilms are multicellular aggregates that sometimes adhere to a surface. Cells that are found within the core of a biofilm have limited access to nutrients and therefore slower metabolic activity. Biofilms are also very highly implicated in persistent infections, so it's really important for me to use them in my experiments. And we see this physiological tolerance of dormant cells when I try to treat cells that have been growing in a static culture as a biofilm for as little as 24 hours. Now, it doesn't matter if you have the beta-lactamase gene or not. A very large percentage, over 40% of the cells, survive a beta-lactam treatment. And I just want to stop here to emphasize that this bar and this bar represent the treatment that we currently give kids for ear infections. And it doesn't work if the cells are in a certain physiological state when you go to treat them. So physiological tolerance is a really big problem that also needs to be addressed. But where does this phenotype come from? Well, one way that cells can become dormant is through a highly conserved pathway in bacteria called the stringent response. It occurs when the cell is starved of amino acids and fatty acids, when the second messenger PPGPP is produced from purines. It halts growth pathways and promotes stress responses. The enzyme REL-A synthesizes PPGPP, and the enzyme SPO-T can both synthesize and hydrolyze PPGPP. The stringent response has not been very well studied in H. flu up until now. I made stringent response knockout mutants in H. flu, and I confirmed them by PCR and whole genome sequencing. First, I knocked out the PPGPP synthetase, REL-A, and this mutant isn't making much PPGPP, but it's still able to break it down. It's also still able to grow as a biofilm. Sadly, I can't make a SPO-T single mutant using the tools that I have currently in my arsenal. PPGPP presumably accumulates in this mutant, and so the cell is placed in a permanent and unculturable state of dormancy. But I was able to make a REL-A SPO-T double mutant, which completely lacks the stringent response pathway, and this mutant was also viable. And so we can see from this data that biofilm formation is not affected by the lack of the stringent response. But next we wanted to know if the tolerance phenotype that biofilms exhibit is affected. And indeed, we see significant killing in both the single and double mutants when we treat with an antibiotic. So in the absence of this pathway, we have less tolerance. And this shows us that the stringent response could be a really good target to inhibit with a drug, because it could also be used as an adjunct along with antibiotics and clavulanate. So next I wanted to know what other phenotypes could be affected by these mutations. Are there other consequences of lacking the stringent response? And is this something we should be targeting with a drug? So earlier I was telling you about competence, which spreads antibiotic resistance. And based on what I showed you about how resistance contributes to persistent infections, that seemed like a really important phenotype to examine in these mutants. So I laid out the pathways side by side, and we noticed that purine precursors, which are used to generate PPGPP, are also involved in regulating the natural competence pathway. So I'll quickly walk you through how the regulation of competence works in H-flu. When a cell is in a nutrient-rich environment, the master regulator of this pathway is repressed. But when preferred sugars are low, cyclic AMP is produced, which activates the master regulator. Then genes that are involved in a starvation response are turned on, and another transcription factor called sexy gets transcribed. However, the translation of sexy mRNA into protein is inhibited by purine precursors. And only when purine precursors are low is the sexy protein produced. So sexy binds to the master regulator, and together they turn on genes that are involved in DNA uptake. So finally we get this apparatus built, and we're able to take up naked DNA in the environment. And here's where we think our connection is, where purine precursors are being drained. When a cell makes PPGPP, it draws from this pool of purine precursors. So I hypothesized that the stringent response mutants, which are not draining this purine precursor pool, they would have a defect in natural competence. And so I took my mutants and I put them through the standard protocol for inducing natural competence experimentally. I take actively dividing cells and I abruptly transfer them to a nutrient-depleted medium called M4. I incubate them in M4 for 100 minutes, after which time they have stopped growing, but they've reached their maximum level of competence. Then I incubate my cultures with genomic H-flu DNA that contains several antibiotic resistance markers. I plate the cultures, and then I calculate the transformation frequency. And I saw that my hypothesis was correct. Both the single and double mutant have significantly lower transformation frequencies compared to the wild type. So this demonstrates that there is some kind of crosstalk happening between the stringent response and competence pathways. Interestingly, we also observed that the double mutant and only the double mutant exhibited a viability defect in the starvation medium. So perhaps the SPO-T enzyme, which is still intact in the single mutant, is compensating for the loss of REL-A. And this result shows us not only that the stringent response supports survival during this rapid change in nutritional availability, but also that the roles of REL-A and SPO-T are separated in H-flu. Because this is in contrast to the antibiotic tolerance data that I showed you, where both mutants were significantly impacted. So clearly there's crosstalk happening between these two pathways, but this result still doesn't explain what regulates it, and so I don't yet know if my proposed mechanism is correct. And lots of different things could be going on. But what I believe is happening is an accumulation of purine precursors in the stringent response mutants. So when we knock out the stringent response enzymes, PPGPP isn't being produced, and the concentration of purine precursors increases. That in turn could lead to the increased repression of the translation of sexy, and in turn decreased expression of all of these downstream genes involved in DNA uptake. But it could have less to do with the simple accumulation of purine precursors and maybe something to do with direct effects of PPGPP. In other organisms, PPGPP directly interacts with enzymes that are involved in purine biosynthesis. And so if this happens in H-flu 2, that would also lead to an accumulation of precursors. PPGPP could also impact the transcription of competence genes in a manner that's independent of sexy, perhaps through an interaction with a different transcription factor. PPGPP could also have direct effects on the function of the DNA uptake machinery. Maybe it's helping to stabilize it somehow. But importantly, none of these alternative mechanisms are mutually exclusive, And so I plan to investigate each of these avenues of regulation. And among other plans, my journey towards uncovering this mechanism brings me first to an RNA sequencing experiment, which I have just recently completed. And if you come to ISOM in June in my home turf of Philadelphia, I will have analyzed the data by then and I can provide you with an update. But the ultimate goal is really to develop a drug that can be administered alongside antibiotics to further disarm the bacteria and make our current arsenal of antibiotics more effective again. And by specifically targeting the stringent response, this drug could prevent physiological tolerance and horizontal gene transfer at the same time. And so that means we're not only treating an infection now, we're treating future infections too. I want to thank you all for listening. I'd like to thank my mentors, Josh and Garth, as well as everyone in the Center for Advanced Microbial Sciences in Drexel University, particularly the names who are bolded. And thank you so much for listening. Thanks to COMMeND for sponsoring my trip here this week.
Fantastic work, Mary.
Thank you.
Questions?
Hi, great talk.
Thanks.
My question is going to be more just because I'm not familiar with Haemophilus, but when it undergoes competence, does Haemophilus, this is so far down here, someone's really short. When Haemophilus integrates DNA, does it make it into plasmids, or does it integrate into the chromosome, and does it have preferred regions that it likes to integrate into?
That's an excellent question. So Haemophilus only recognizes DNA that has a very specific uptake signal sequence. So there's a specific sequence that it recognizes in the extracellular DNA. It gets inserted, and in the process of doing so, the DNA gets split into single strands. And so that single strand of DNA can eventually get incorporated into any part of the genome that through homologous recombination it matches up with. So yeah, it can get inserted anywhere.
Oh, excellent. And is there a particular length that it will take in, or will take any length of DNA, or you prefer particular sizes that's integrated into? Now I'm talking just from my experience with strep pneumonia. Strep pneumonia is very similar, but it does like a certain length of DNA that it will take in. So, you know,
Yeah, that's a really good question. I'm not exactly sure of the exact size, but I have given each flu just genomic DNA, like as a plasmid, and it's taken it up before, so yeah.
Awesome, thank you.
I have a quick question for you. You proposed using a stringency inhibitor to increase antibiotic effectiveness. What about the gut bacteria? Do they have a stringency response, and might we be making that problem worse?
That's a really good question. I know that there are stringent response inhibitor drugs that are currently like on the market that are being used as adjuncts with antibiotics. I haven't heard of any issues with like causing issues with the gut microbiota when taking that drug, but I imagine it would be sort of similar to the use of an antibiotic. Like if you use too much or you're on it for too long, it will cause problems. So there's definitely potential there, and that's something we'll have to consider when we are finally at the stage where we can make this into something that would be a deliverable product. Yeah, but that's a really great point. Thank you.
Well, thank you very much.
Thank you.
Appreciate that talk. Our next speaker is Evangeline Williams, and she's going to talk to us about clinical bacterial transcriptomes, which I think is going to be fun.
All right, good evening, everyone, because now we're in the evening. My name is Evangeline Williams. I go by Leanie, and today I will be talking to you guys about the work that I've been doing throughout my PhD. And so, as we've been mentioning, otitis media is the number one reason why children go to the doctor, are prescribed antibiotics, and receive surgery. And about 3/4 of all children will have at least one episode of OM before they're 3 years of age. And most of these acute infections resolve on their own or with antibiotic treatment. However, up to 1/4 of these infections will continue to go on to become current or chronic or recurrent episodes of otitis media. And this occurs at a critical point in time where these children are learning language and is important for language development in the future. And most of these infections are due to Haemophilus influenza or H flu. H flu is present in the nasopharynx of healthy children as a commensal organism. However, it also has a high burden of disease. It's the number one cause of sinusitis and conjunctivitis, as well as these chronic infections such as otitis media, as well as lung infections and chronic obstructive pulmonary disease. And many of these chronic and recurrent infections have a biofilm component. And biofilms are aggregates of metabolically dormant cells surrounded in a matrix of extrapolymeric substances produced by both the microbes and the host. These biofilms protect the cells from the host immune system, from opsonization and phagocytosis, as they're too large. Additionally, the cells within the biofilms are metabolically quiescent, as they are unable to receive as many nutrients and oxygen. And this environmental stress leads to having a high antibiotic tolerance. Antibiotics, as you know, target growth processes such as DNA replication and protein synthesis. This results in cells in the biofilms being 100 to 1,000 times less susceptible to antibiotic treatment when compared to planktonic cells. And this plot here shows that, excuse me, across all classes of antibiotics tested, cells that have been starved for at least 24 hours are between 5 and 70% less susceptible to this antibiotic treatment. And this is just a single type strain that we were previously looking at. So it is important that if a lab adapted strain can withstand up to 250 times the minimum inhibitory concentration of antibiotics for the first line treatment of OM, then it is essential that we learn how to detect and treat these biofilm infections. However, we also have to ensure that we address the formation of biofilms across diverse clinical isolates. Now, Haemophilus influenza has high genomic diversity. What you see in the center here, we can visualize this genetic diversity by making a pan-genome matrix of all the genes that are core and distributed across a selection of strains. And what we see is that up to 80% of these genes are shared among these strains. If we then take a subsample of only 10 strains and plot phenotypes such as growth rates or biofilm biomass, we see a wide range of variation among these strains. So how do we know that the mechanism behind this biofilm formation will be the same if these phenotypes are not? So we used a collection of strains that were collected from our collaborators in the Pekikaro lab. And these specimens came from children who were either healthy or had delayed innate immune development. And these children went in for routine pediatric visits and either received a nasal pharyngeal wash or the middle ear fluid was collected from an episode of acute otitis media. And we used this collection of specimen as a representation of the species. And so what we have learned is that we can use only 9 of these clinical strains as a subset as a representation of this genetic diversity. Of these 9 strains, we can capture all the accessory genes that are found at a frequency of greater than 10%. These strains are also diverse at the nucleotide level, which is represented by the distribution of these colors on the phylogeny. Additionally, we've included 3 well-studied type strains in our what we've affectionately named our diversity panel. And then these 12 strains we've been using as this representation of the population. So throughout my work here, I've been using this diversity panel to understand biofilm formation. I have been aiming to identify shared patterns of gene expression, which represent this biofilm formation across the species. I additionally am using these strains to identify diverged patterns of gene expression, which can distinguish a good biofilm from a bad biofilm. We've performed a large-scale RNA-seq experiment to measure the gene transcript abundance and production over this biofilm development. And this included creating a Haemophilus influenza pan-genome to use as our reference for counting transcript abundance. RNA was extracted from 5 conditions for each of the 12 strains in triplicate. And because I aim to identify the common biofilm formation signatures, I first have been examining only the 1,263 genes that are core to our diversity panel. And to visualize these transcriptomes, I start by making a principal component analysis, or PCA, which represents how similar each of the samples are to each other. Each point represents a sample, so there are 173 dots on this plot. And what we see first is that the transcriptomes cluster by growth condition. Each color represents a time point from exponential phase, where the cells are happy and growing with high amounts of nutrients and oxygen, to a 24-hour biofilm where they've been starved. And additionally, within each of these conditions, these replicates of the same strain cluster together. And examining the differentially expressed genes for just 2 of these strains, we see patterns of genes that are up and down regulated in the orange and green respectively. Patterns of gene expression that are similar among these conditions. However, the strain background significantly impacts how these genes are expressed. We then use functional analysis to examine the pathways which change. And we see that regardless of strain background, the transcripts, the strains are increasing expression of protein turnover and transcription while downregulating translation. And because we know that these transcriptomes cluster by condition, and then within each condition, these strains are clustered, we can then start to identify these diverged patterns of gene expressions between any strains. And I don't exactly want to go over every single differentially expressed gene for each of the 12 strains, but we can examine these transcriptional differences, and this allows us to ask if any of these transcriptional differences are correlated with a biofilm phenotype of interest, such as biofilm biomass. And although each strain has hundreds of differentially expressed genes, we can take advantage of bacterial biology to reduce these gene sets into manageable sizes. We know that bacterial genes are co-regulated as they're arranged in operons, and promoters control genes with similar functions. So how can we reduce these hundreds of genes into a network of predictive biomarkers? We've used weighted gene correlation network analysis, or WGCNA, to cluster the 1263 genes that are core to the diversity panel into only 32 modules of correlated expression. These modules range in size, and module expression can be estimated by a singular hub gene. The hub genes have the most interactions between and within modules, and often have important biological functions. And in our preliminary analyses, we see that this module expression can be correlated with the phenotypic traits. For example, 10 of those 32 modules expressions in an early biofilm time point are significantly positively or negatively correlated with the biofilm biomass at 24 hours. Our next step is that we could use combinations of these modules expression to correlate with these predictive traits. Additionally, we can begin to test our biomarkers on additional strains within our collection to see if these biomarkers hold true. And then additionally, we can examine other biofilm phenotypes such as ATP levels or growth rates in different pHs. And so what I hope you guys can take away is that bacterial genetic diversity complicates these functional analyses and diagnostics, but it also represents an opportunity for us to understand this phenotypic diversity. And eventually this could lead to diagnostics of persistent infection risk. And so I would like to thank everyone who has helped me with this work and has helped make this possible. My thesis committee and my PI, Dr. Josh Mell, as well as the other member of our lab, Mary Marino, the rest of the Center for Advanced Microbial Processing, Drexel Microbiology, Immunology, COMMeND for giving me the opportunity to present this work, as well as the families that have been kind enough to donate their specimen for me to study. And I'd like to take any questions. Oh, also go birds.
All right, one question already.
This is the perfect time. Anyway, so thank you for your research. We actually looked at similarly in chronic subarachnoiditis media clinical samples, looking at multi-species bacteria that were Staph or Pseudomonas Haemophilus. We found that the NYCs and the bioconforming capacities were, and persistent cell capacity was associated. They were completely independent. So it'd be interesting to see, find that there is a correlation going forward. I always wonder, did they become like this through evolution, or did you pressure this through antibiotic exposure over time? And is there a way that, since you're looking at the genes, a correlate could be that a lot of these are starting to upregulate multi-drug efflux pump genes as they're becoming exposed to antibiotics, whether that's a correlation we can look at?
Yeah, that's both interesting comment and question. So kind of the purpose of our study was to avoid studying every single strain as much as possible and then just reduce it down into ones that we could use to represent it. And what we specifically knew is that because we've reduced down to our diversity panel, we've taken the ability to correlate with any of these phenotypes out. So we were avoiding doing the genome-wide association study. But my goal would be to see if we could actually expand it to other phenotypes, such as these persistent risks, knowing that there are so many of these complex interactions between other strains, polymicrobial, polyclonal infections.
Awesome, love biofilms. Really good talk. I'm really interested in your results as well. I don't know how long I can stand here on my typical, so I'll make it quick. I'm wondering if you could tell me a little bit more about your biofilm model, like what media you used, what conditions you used, and how you measured.
So we're using a 24-hour well-associated biofilm. We're mostly calling it this starved biofilm, in that we're not using the perpetual flow models, because we are seeing these transcriptional changes as early as these 5-hour biofilms. But it is mostly due to this starvation. We're just using supplemented BHI, and then just enough leaving them for 5 hours, we do see these changes. We have looked into using the the flow reactors for the biofilms, but we have to reduce the time down to only a single 24 hours because we go through so much media. So using the just a single 24 or 48 well plate in the starved 24-hour conditions.
Yeah, I wonder if you have any plans to incorporate some cells in there and whether you might see any differences in expression there, and also if you find any niche specific expression depending on which cells you use.
I mean, I only have so much time in my PhD. We have seen a lot of these. A previous student had done co-culture with for Moraxella, and I'm not sure what host cell she was using, but yes, we have previously done a lot of these co-cultures, but not recently.
Yeah, cool. I'm doing some biofield work at the moment, and I always wonder, it's always easy to throw things in a plastic plate, not have to worry about the cells. So I'd love to know if there's actually any differences there, and we can just get rid of the cells up and much.
Any other questions? If not, thank you so much. That was a fascinating and very beautiful presentation.
Thank you.
Thank you.
Right, last but not least, we have Dr. Guillermo Monroy from the University of Illinois Urbana-Champaign. We will talk about the novel treatment for otitis media using cold mycoplasma.
All right, thanks for the introduction. I've been working on this project for about 2 years, and so I want to kind of show some of the kind of overall results I've had so far. And it's been great listening to the other talks. I think there's a lot of points to learn and other ways I can analyze this project. So I want to first acknowledge my group and the many collaborators here that made this happen, kind of underlined and highlighted there. I'm at the center of this, but I've had people train me in MRI imaging, ABR. I'm coming from the engineering side, so this is all a little new to me. I don't think I need to go over the importance of diagnosing and treating otitis, so I'm going to maybe skip over these and save some time. But I think it's very clear how many infections there are a year, how often antibiotics are prescribed, and how many surgeries there are per year, and really just how much time this all takes. Of course, for treatment, which is what we're going to be talking about today, we have watchful waiting, multiple and different types of antibiotics, and then of course surgical placement of tympanostomy tubes. So I want to focus on antibiotics, and really the point I want to drive home here is that they're very effective. So I'm not going to throw away years of research that these are somehow now today all of a sudden not useful, but we just don't get anything for free. So we get this effective treatment, but then we have risk of antibiotic resistance. We can change the microbiome, as we're learning from those folks in that research field. Children that are exposed to antibiotics for otitis primarily then end up with adult-like microbiomes when they're about 3 or 5. And then there, of course, can be drug shortages, and so when that happens, then you really have to change your strategy. So again, I'm coming from engineering, and there really isn't a strong alternative to antibiotics yet. And so I'm always thinking, how can I do better and kind of improve healthcare? And so I hope this is kind of one push towards that. I spent the majority of my PhD developing OCT, optical coherent tomography imaging for diagnosing otitis. And so this is much like optical ultrasound. You can look at the middle ear cavity, anything kind of near the eardrum itself. And so in this presentation, I'm just kind of flipping these for convenience. But then you're able to really look inside the middle ear cavity and really kind of look at the entire progression of someone's infection and kind of stage where they are depending on when you image them. And so there's tons of studies. I really encourage you to go look at this if you're not familiar with this work, for myself, for my collaborators and colleagues. But today I really want to talk about plasma medicine. Plasma is typically used in semiconductor manufacturing. Some groups are starting to use this now to sterilize things, so for medical devices, water and exhaust gases, fruits and vegetables. Other groups still are finally getting into plasma medicine now, finally. We're looking at reducing pathogenic load in wound beds. That's in Europe. And this company, USMI, just got a FDA approval in, I believe, last year in October to use this for tumor resection therapy. Once you remove the tumor, you blast the cavity with plasma to try and reduce any unwanted cancer cells you'll leave behind. I'm going to apply this to otitis, and there's a lot of mechanisms I can use to impact that. I'm primarily going to be relying on the generation of reactive oxygen and nitrogen species to cause oxidative stress to wipe out the bacteria. And you might be thinking, oh gosh, this is a terrible idea. Let's see if I can get the mouse over here. And so if you think about the body as a system, we have a lot of, I guess, like sophisticated and robust methods to manage oxidative stress. I can list these off, but they're there on the screen. And the bacteria kind of lack that robust strength, but also then the kind of level of resistance to them. So I think there's a Goldilocks zone that you can get into where you're not causing damage to the host cells, but you can clear out all the bacteria. So this is kind of the idea. To generate plasma, you run specific noble gases through ionized or high voltage plates to ionize the gas. And then that generates a plasma, which then downstream generates these reactive species. And that will pass the reactive species through the ear canal and through the eardrum into the middle ear cavity and to treat that infection. So there's been a lot of work in the past in vitro. So we've already tested planktonic and biofilm susceptibility just in a dish. And that reducing line there, I'm not going to rely on the mouse here too much. That reducing line, all that means is that you need less and less antibiotics to clear the infection in that dish. So with more plasma exposure, there's less antibiotics needed to get beyond that MIC level. We also made sure that this plasma device can pass its radicals through an eardrum. So we used a rat eardrum ex vivo. But of course, if that somehow blocked gas diffusion, that would kind of stop us right there. So that was still effective through that. There is some loss, of course, but enough of it does get through to be effective on the other side. And then finally, maybe most importantly, the course viability testing was done. So it's fine if we can clear, just like hand soap, right? 99% of bacteria, but if that 1% survives and regrows, then it's kind of pointless. So we did also confirm recently that the bacteria are no longer viable after treatment, even if they're still alive. So as long as our treatment gets there, then we can really eradicate them. So okay, I wanted to also highlight that this whole project was kind of done from scratch. So this was over a few years. Again, many hands have helped and contributed to this, so I want to acknowledge that here, all the way from the animal housing, all the way to the behavioral assessment and physical assessment at the end. So we're basing this model on a chinchilla model gifted from the Medical College of Wisconsin. Me and some other lab mates went up there and they trained us on how to handle the animals, inoculate the animals using human-derived NTHI. And chinchilla is a really good animal model because they not only are pretty docile and easy to work with, but they also have really reliable and kind of consistent reactions to NTHI inoculation. They can also have really standard antibiotic responses, so they're a well-studied model. Their eardrums are also easy to access the bulla too on the back of the skull, though they're pretty large. Okay, so I think you kind of take for granted that we have this whole antibiotic pipeline now available clinically. We know the dosage, the interval, the frequency of all these things. But with this approach, I have to start from scratch. Plasma has never been delivered into the ear this close to all these fine structures of the brain. And so I have to kind of start and figure out at a base level what's safe, what's even possible for exposure. And so to do that, I'm going to use OCT, otoscopy, and a physical exam. And when I say that, it's what you would normally do for a human, but adjusted for a chinchilla. So I'm looking at a bunch of veterinary factors, behavioral exam, weight, all these kind of things. And so just again, crudely, I'm going to have that plasma device just sitting kind of outside of their ear. There's a guidance tube and an otoscope tip that helps flow that into the middle ear cavity. Okay, so as I showed you earlier, plasma can kind of be run in ablation mode, which you would want on a semiconductor silicon wafer. If we ran that in the middle ear, we would get rid of the bacteria, but we'd also get rid of the ear. So you definitely want to have that running at kind of a minor level. And if we looked pre-post with OCT what the ear looked like, it would look exactly the same, right? We're relying on this kind of biochemical impact to clear the infection. And so within 12 to 24 hours, you really see the properties of that effusion change. So remember there's, let's get the mouse back here. There we go. You get the eardrum here. We're looking at the ear canal, eardrum, and then inside in the middle ear cavity. The properties of this effusion substantially change, and you can quantify that in the image. But really what this is reflecting is a change in the effusion in the microenvironment there. So you're effectively disintegrating all the bacteria, their signaling particles, the mucin, and then you're also stimulating the immune system a little bit. But generally, if much like an antibiotic, if you don't have the right course of treatment, the infection will just come back almost like you never did anything. So really what I had to do, and this took quite a while, is to figure out which parameters in this space matter, which ones I could kind of let go, which ones were informed by others, and figure out how to just reduce this hyperparameter space. I listed maybe 8 or so here. There's about 20 I was kind of looking at, both on the device side, on the therapy delivery side, and then the animal model. But really then the temperature of the device really set everything else from backwards. So if we run the device to be at body temperature or lower, that sets limits on flow rate and power we're dumping in with the plasma, the ear canal size and dimension, and then the risks of hearing loss for gas flow being too loud, that kind of sets the flow rates. So you can kind of see how I narrowed that down a little bit. And over time, I just really was doing this very methodically because, again, I wasn't sure about that a little bit. The main one from this point is that there was dryness and itching in the ear canal itself just from all the insertions from the devices, the otoscope, the OCT system, the device itself. So using some pure lube just kind of takes care of that, no problem. Okay, as I mentioned, I was thinking a lot about, not only for the animal, for myself, I'm sitting basically in front of the animal the whole time while they're being treated and exposed to plasma. So there's high voltage concerns with that plate in the device. There's cytotoxicity issues for me and the animal for being exposed to that constantly. So if you of course, crank this up really high and expose everything to too much reactive species, you will cause tissue necrosis. So I was very careful, kind of coming from 0 up to a therapeutically effective dose. We can run this in ablation mode, which I wasn't going to do, so setting that helped a lot. And then, as I mentioned, so gas flow rates really set at the ceiling again on hearing safety for that at about 70 or 65 dB. I forget exactly the moment. So for about 15 minutes, that's still safe. Okay, so side effects. So again, we're looking at the eardrum with the ear canal in the middle ear behind, below it. And this is in the control animal that wasn't infected to really just see what plasma exposure would do to it in general. And so you can see the eardrum typically is like 50 or 70 microns thick in a chinchilla, depending on where on the eardrum itself you look. With exposure, then you get a little thickening. There's a little bit of earwax over here too, so don't look too hard in that section. But there's about a 30% increase in thickness here, and that's really just interstitial fluid, right, coming in from inflammation. This is tolerated really well. It goes away in a few days, and there's no change in behavior or anything. But I think there's a lot of like immunological things we can look at with this as I've been here and just thinking generally. And you can see in otoscopy too, the ear canal does get more inflamed just because there's more vascularization, right? So I've been considering how to kind of provide a pre or post treatment to kind of cushion the tissue. That'll also change the plasma dynamics, so that's a tricky one to think about. In terms of validation, I also looked at high field strength MRI, and so with Dr. Majumdar there, we were looking at some of the chinchillas, and to give you an idea, we're looking at the back part of the bulla here so that you can see into their middle ear cavities, of course, and so it kind of looks like a kidney bean. You can really see, for example, in this one on the left here, the active infection. You can kind of outline in this slice the middle ear cavity. There's a little fluid bubble at the top, and then there's actually, I think, some biofilm residually precipitated out of the bottom. There's some kind of biomass. And if you compare that to one animal that was inoculated and then cleared, not only are all the middle ear structures intact, the fluid's all gone, of course, but the animal's hearing was intact and everything looked good on that side. In terms of ABRs, again, I'm a novice at this, but I really didn't notice any major shifts in the animal's hearing acuity on this test. Again, maybe some of the subtle changes that happen are going to be missed with this, which is a good fact for me to know. But I'm really looking for, intrusion of these reactive species into the inner ear, which would probably immediately cause hearing loss, but I don't think that would happen. I would have seen it by now. The changes I really saw were more like measurement noise, I think, because again, new with this, placing the electrodes in the right place so that their fur is pretty thick, making sure the experimental setup didn't change how I'm borrowing a collaborator setup, so it's hard to keep track of everything. But generally, I think these ABR tests confirmed that there weren't any major changes in hearing acuity. So there are some limitations, of course, and other than doing more animals, the treatment really just needs to reach the infection. And so we're relying on these reactive species, basically gas, to diffuse to where the bacteria are and affect them. And so in the chinchilla middle ear cavity, like I said, it's a kidney bean, so if we're treating top down, the ear pin is kind of out of the top, we're putting the tube in, and then treating kind of down into that lower section, that might be really difficult, especially if there's a really thick fusion of mucus. And so anatomy is going to have a large impact. In a human, actually, which is unusual, usually the human case is much more difficult. The anatomy just kind of helps us out of it because then we can kind of bubble in from the bottom up. The other piece I mentioned, we talk about later, so bacterial load has a big effect on this, of course. So with antibiotics, again, we're taking for granted that that dose will
will allow you to treat the infection successfully. So since this is a device, we can kind of run it as long or as little as we want. And so maybe there's a prevention level 1 for acute infections, level 2 for chronic infections. That's something we can explore later now that we have this kind of base protocol set. Because before, I think we weren't sure kind of what protocol would work. So that's definitely an optimization. Okay, so this is kind of the idea. If we're going to go into humans, there's a little earbud or some kind of otoscope-like device, and the point would be to pop this into a kid's ear. Maybe they can just sit and watch their tablet for a little while, and then go home and be treated. But really, that would require many years of study and a complete reimagining of that treatment pipeline. So realistically, kind of in my lifetime or career, I think a more realistic path would just be to change and maybe reduce the antibiotic exposure course for that, and kind of optimize that. And really, if you think about why that's possible and effective, if you think about the whole kind of pipeline that children follow, like the worst of the worst cases, where there's watchful waiting, let's see if we can do this here. Watchful waiting, a trip to the primary care office, maybe the ENT through surgery. I'm providing what I call exit avenues, where maybe you prevent or just disrupt the biofilm from being established or surviving. Reducing the risk of antimicrobial resistance because you're giving just a lower dose or a shorter course, maybe preventing the need for that strong secondary exposure in the first place, or even reducing surgical burden by either preventing people from getting to that point, or just using this surgery when the eardrum's already open. Because that's one thing for this is, it's all been non-invasive to this point. So really just kind of interrupting this, endless cycle that people face over and over to then really change the way care is delivered for otitis. Okay, so I know I covered a lot. Please reach out with questions over email if you want, or I'm here to take questions now. Thank you.
Thanks, Mark. We are